see. And so we're a little ahead of the broadcast. Just waiting for it to pop up on my other screen. Okay. Well, it looks like we are hit the noon hour. So welcome everybody. We're going to go ahead and get started. Good afternoon. Welcome to the Lunchtime Lecture Series from the Botanical Research Institute of Texas and Fort Worth Botanic Garden. I'm Dr. Brooke Best. I'm Director of Research Programs at BRIT, and I and my research colleagues oversee this lecture series, which takes place the first Tuesday of each month. So before we get started, I want to mention that this lecture and the accompanying chat in YouTube are being recorded for archival purposes, and they will be available in the future on BRIT's YouTube channel, pending permission, of course, from the speaker and their organization. Links to the video and any other resources, if needed during the talk, if things are shared and we want to post a link somewhere, we'll probably put it in the chat, but we'll also, those will be available after the talk on the BRIT event page for this event on BRIT's website. Okay, so I'd like to present our guest speaker today. So we're delighted to have Dr. Dori Contreras. Dori joins us from the Perot Museum of Nature and Science in Dallas. Dr. Contreras is a curator of paleobotany who's been with the Perot since September of 2019. Dori got her bachelor's from Texas State University in San Marcos, Texas, and did her PhD out in California at the University of California, Berkeley. Her paleontological fieldwork has taken her from New Mexico, California, Argentina, and now back to Texas. So without further ado, Dr. Dori Contreras presenting her lunchtime lecture today. Take it away, Dori.
Great. Thank you so much, Brooke. It's really great to be here today, and I want to thank you all for showing up and coming to this talk. I won't be presenting on the entire history of fossil plants and the plant record today, but I will be keying into just one really important and I think fascinating time in plant evolution, and that's when flowering plants took over the world. And I want to do this talk in basically two parts. The first one, I'll kind of summarize what we know about this time in plant evolution, kind of going over the major trends and patterns and just kind of take a walk through that. And then for the second part of the talk, we'll take a little pivot, more of a deep dive, looking at some research that I started during my dissertation and is ongoing and I'm continuing today, looking at this really fun and incredible flora from South Central New Mexico and how what we're learning from that fits into this broader picture of understanding this time when flowering plants took over. So let's get started. First, I want to start by taking a step back and thinking about our modern world and the vegetation that exists on our planet today. So if you look at kind of across the globe, we have a bunch of different types of forest and floras. From boreal forest, we have our temperate forest, tropical rainforest, subtropical forest. Here in DFW, we have our lovely prairies. And of course, all of these, or most of them, at least with the exception of places at high latitude and high altitudes that are dominated by conifers, are pretty much completely dominated by flowering plants or angiosperms. And that's in terms of diversity. Flowering plants are the most diverse of all the plants, but also in terms of growth habits and ecology. So our flowering plants do everything from being herbs to small shrubs to absolutely giant canopy trees to lianas, vines, epiphytes, just a ton of different ecologies and growth habits. And they just plain dominate. And that's really interesting to me because, relatively speaking, flowering plants are the new kids on the block. They're the land plant group that evolved most recently. So let's take a little look at that history real quick. So if we look at the timeline of kind of the major plant groups and when they evolved, here's a timeline going back over 400 million years and all the way to a lot more recent. And on the x-axis, we have the number of fossil species just kind of tabulated through time and broken out by our major plant groups. And we start out with these kind of early land plants. We have some free-sporing vascular plants, things like ferns and lycopods came around. Then seeds evolved, and we have gymnosperms. This is our non-flowering seed plants, like conifers and cycads and a whole bunch of extinct, super cool seed plants. And that's what existed and dominated the world before the flowering plants came around. So in the Jurassic here, when dinosaurs were already around, we pretty much had this gymnosperm fern world. So conifers, cycads, ferns, that's what the vegetation mainly consisted of. And then in the Cretaceous, flowering plants show up in the record, and they just absolutely take off. And you can see that by looking at this. They take off and become really diverse, and then eventually we're here at today when flowering plants are everywhere. So the Cretaceous, this period, is kind of what I consider to be the dawn of modern forests. This is when things really hit the trajectory towards the vegetation that we see today. And it happened really rapidly. This was a fast takeover. From once they appear, by the end of the Cretaceous, we have all the major groups or clades of flowering plants have already appeared. And right here at the end of the Cretaceous, when the dinos go extinct, right afterwards, we have rainforest in Colorado, angiosperm-dominated rainforest. And you might say, wait a minute, did you just say Colorado, rainforest in Colorado? And yes, this was also a very different world. Angiosperms evolved when the world was a lot warmer and the continents looked, well, they were configured a lot differently than they are today. So here's the end of the Jurassic, right when angiosperms are arriving on scene. And you can see, well, the continents look a lot different. We have no ice caps on the poles. Sea levels are higher. This is a warm time in Earth's history. And as we progress through the Cretaceous, things change more. We have the breakup of Gondwana. The Atlantic Ocean here opens up. We have these shallow seas that cover the interiors of many continents, like here through the US. This is the Western Interior Seaway. If you happen to explore a lot of the creeks and things around the Dallas-Fort Worth area, you might find lots of fossil shells, oysters, things like that. And that's from this time period when seas covered this area. But it was much different. So warm world. And that persisted all the way through the end of the Cretaceous, and we had this lovely asteroid just hit the Yucatan and just mess things up. So point being, angiosperms evolved and spread across the globe in this very different environment. Okay, so now we're going to zoom in just on the Cretaceous. This is our key time period, and I want to kind of go over just these main trends throughout the Cretaceous in kind of what happened with angiosperms. And we'll start with the story from fossil pollen. So this is the pollen, the same kind of pollen we think about that is carried around by bees and the wind and covers your car and gets in your nose and wreaks havoc on allergies. Well, pollen is so ubiquitous, it settles into the sediments in the soil and it enters the fossil record. And pollen's really tough, so it remains there. As a result, we have this really incredible fossil record of pollen that, compared to other records, like of bigger fossils, is really continuous and fairly complete. So it's a really great resource for knowing where and when things happen without really a whole lot of major gaps due to preservation. So it's not surprising with how good pollen's fossil record is that the first evidence we see for angiosperms in the fossil record is pollen. And that shows up about 137 million years ago in the early Cretaceous. And it shows up as these kind of early, we call them simpler types of angiosperm pollen called monosulcates. And you don't need to remember these names, but there's just two types, these monosulcates and then later tricolpates. And there's a big diversity of them. These are just a few diagrams of those early forms of angiosperm pollen types. but they show up here. We do see leaves in the record a little later, flowers a little bit later. They're just more rare in the fossil record, so we don't see them quite as early as we see pollen. Now, once we see the pollen, we have this really interesting pattern of spread across the globe that's told by the pollen record. Here we have 4 maps. There's A, B, C, D of the globe, and these kind of track through time, A, B, C, and D. So youngest here to oldest here. And these plot out different places in the world that we have pollen records. That's what all these little circles are, little samples of pollen, fossil pollen. The ones that are orange are places where in those samples there's flowering plant pollen, angiosperm pollen. In places in the circles that are white are places where we have a pollen record, but there's no angiosperm pollen. What these tell us is that when angiosperm pollen first shows up, it shows up in the equatorial region. So that's where they first appear, is around the equator. And they appear as these kind of more primitive forms, the monosulcates. As we go through time, the angiosperms spread towards the poles, interestingly, towards the southern hemisphere first. And some of those tricolpates, the more advanced form, shows up in the equatorial region. Then those spread out too, and eventually, by the late Albian, so that's up here at the end of the early Cretaceous, angiosperms have spread to both poles. They've kind of now got a worldwide distribution after starting from these equatorial regions. Now, at the same time that they're spreading towards the poles, they're actually getting more diverse in each of those places. So here's a plot looking at within each of those like little dots we saw on the last maps. If you counted out the percentage of pollen species in each one of those records, that's flowering plants versus other plants, and plotted them, this is what you get, kind of this record through time of how many angiosperms we see in each sample. And what you see is that really fast upward trajectory. That shows us that at each site, angiosperms are diversifying. They're becoming more and more diverse at each site. Basically, by about 100 million years, where this line is here, angiosperms are making up more than 50% of the species at many of these sites. That's right here on this timeline. So by here, we know from the pollen record that we have really high species richness, so that's diversity, of angiosperms in floras across the globe. So that was a pretty quick trajectory to becoming really diverse everywhere. Okay, now the pollen record told us about the where and the when and gave us a really good broad overview. But if we want to know what the ecology of these plants are, what do these plants look like? What are they doing? Then we turn to the macrofossil record. And a macrofossil is just like, think of it as a big fossil, right? These are things that we don't have to use microscopes to necessarily see. And so here's kind of just a random sampling, some eye candy of some of the early Cretaceous angiosperms that are known. There's many, so here's just a sample. But what we know about these early Cretaceous angiosperms is that for the most part, they appear to be these fast-growing herbaceous plants. You can kind of think of them as a weedy habit if you want. And that was kind of the primary ecology we see. They tended to live in disturbed environments, like channel margins or these kind of shifty places. And we know that by their association with the sediments and the rock layers that they're preserved in. So this is a really beautiful reconstruction of a flora known from the Far East Russia about 110 million years ago, so about right here. And here in the foreground, along this channel margin, this disturbed environment, you see a lot of herbaceous angiosperms. There's some ferns in there too. When you get outside of these channel margins, it's pretty much mostly ferns. And when you get into kind of further away from the banks, off into what we call the floodplain and in these upland areas, it's still mostly conifers. And this is kind of exemplary of a pattern that we see throughout the early Cretaceous, that angiosperms are along channel margins and places like that. And then outside of those, on the floodplains and upland areas, it's still conifers, gymnosperms, things like that. Okay, now further exploring the macrofossil record, we get into woods. So we do see a lot of permineralized or petrified woods. You can cut them open, slice them open, look at the anatomy, and determine what that plant was. And if you look at there is a nice fossil record of flowering plants as permineralized logs and stumps throughout the Cretaceous. For most of the early Cretaceous, there's only a few samples. So this is kind of a plot through time of the woods and logs of angiosperms that are found, plotted by their diameter. So down here, they're really small. They're pretty sparse through the early Cretaceous. And then they start getting bigger. So we start seeing bigger and bigger logs. This was one found kind of from this age here in the Turonian. It was surprisingly big when this was found just a few years ago for the age. So this is its star up here. We'll actually visit this big old one later in the talk. But all in all, we see this trend as we get into the late Cretaceous where angiosperms are clearly getting bigger. They're forming trees. We know that by here there was some big angiosperm trees, and they get a bit bigger through the rest of the Cretaceous. So we're starting to see the growth habits kind of diversify and really starting to enter the canopy and not just be these more herbaceous, smaller things. Now, what's interesting about that is even though we know that they're forming some trees, when we look at kind of the composition of floras, what we've kind of overall taken from it is that angiosperms don't seem to have gained widespread dominance by abundance, so being the most common thing across a landscape outside of channel margins, until the very end of the Cretaceous. And that was an interesting pattern considering they're really, really diverse in floras way back here at 100 million years. And for them to not be really common across broad landscapes until 65 million years ago. So this kind of discrepancy between when they're diverse and when they're abundant has been called this ecological lag. So it's kind of the ecology dominance is lagging behind the diversity dominance. And this is kind of extended to even kind of lagging and thinking about what was their role in canopy and what diversity of growth habits did they have during this time? When did they evolve all the traits that made them be able to do all of these different things and photosynthesize so fast? So this was clearly an important time, and we have a lot to learn about it. This is kind of the time period that I find to be really most interesting. This is when we still had this transition from the gymnosperm fern floras to angiosperm dominated floras. So this shift. And this is what we're kind of gonna hone into for now. And then there's a lot of questions that that I have and many people are investigating about that period. First of all, during that transitional time, how did these different plant groups contribute to the structure of forest? What were conifers doing? What were angiosperms doing? How did they mix together? What did these floras look like? And then kind of stepping back more broadly, what were the pattern of change in Cretaceous communities? When and where did angiosperms really start taking over? And then kind of extending further, I mean, we have to remember that this was also a very different world. We had dinosaurs tramping around these forests. This was a much different environment. And it's interesting to think about how angiosperms really contributed to these changing ecosystems. How did they interact with dinosaurs? What was the effect on the insects? There's many questions there. Okay, so we're gonna hone in mainly on these first two questions. And we're gonna do that looking at really one place in time, and that's here, the western interior of North America. And we're gonna do that because it has a really incredible fossil record. Because of all this geologic activity, this interior seaway, we have a lot of floras preserved. And so we're learning a lot from it. So here is a cartoon version of that box, the western interior seaway. Here's us in Dallas, sitting in the middle of the sea. And we have this really incredible record of floras. So it's all these little orange dots kind of spanning this margin of the seaway. What we know from studies of these floras is that at this time, there was still a gradient in flowering plant diversity in the species composition of floras, to where angiosperms flowering plants were more prevalent at the lower latitudes. So kind of consistent with that latitudinal spread we saw from the pollen record, just extending later into the Cretaceous as well. Now, we know a lot about diversity from these floras, but only a few of these records have actually been studied in a way that we can get at community structure, get at these landscape reconstructions. And that's really only 3 floras so far have started to be counted and studied in a way we can look at relative abundance and things like that. And that's the Big Cedar Ridge flora here in Wyoming, the Kyparowitz and the Fruitland-Kirtland here. These are kind of below what this tropical thermal belt line is, where we start getting into really tropical temperatures at the time. And these studies are ongoing. There's some preliminary data out on them. But this flora up here, I want to take a minute to look at because it's a really incredible record. And this study that was done on this flora has been really influential for the way that we understand vegetation here in the Lake Campanian during this time. So let's look at that. So here's some gorgeous reconstructions of the Big Cedar Ridge flora. It's a 73 million year old flora. And basically what it is there was a giant ash flow, kind of a mud flow, if you will, made of ash from volcanic activity that flowed across this landscape, buried it as it was, and then that got preserved. And a group of researchers, came across this. It turned out this deposit was 4 kilometers long. So they basically have a 4 kilometer long snapshot of a flora from the Lake Campanian. And they did an incredible amount of work on this over, oh my goodness, I think it was 2 decades. They dug 100 quarries. That's 100 big holes in the ground across this 4 kilometers. And they counted, identified, and described everything they found. In a nutshell, what they learned from that was that flowering plants were really diverse. They were the most diverse thing, as we would really expect. But surprisingly, ferns were the most abundant thing. Once they counted the relative abundance of all the species, it was ferns that were most common. And this was pretty surprising. It turned out it was a palmetto fern wetland. And the only place that angiosperms were actually kind of abundant was in the streamside vegetation. So kind of these patterns that I discussed a little earlier. So this was surprising because we're like, wow, they're so diverse. But here, even at this time, this is a landscape still dominated by ferns. Now in these other 2 floras, we're kind of seeing similar patterns. Flowering plants are really, really diverse, and they're really diverse in these channel environments. But we still see in uplands and things like that, a lot of conifers. Now this is just a few dots, and this is where I'm going to get into talking about the research that I've been doing. I'm adding to this record, looking at a new flora that's along this gradient, but further south. This is from the Jose Creek member of the McRae Formation. This flora seemed like a really good place to study, to look at these patterns, given that it's, well, further south, but it's also more inland than most of these other floras. So most of the fossil record that we have is preserved along the seaway, kind of in this light green area. This is the coastal plain. This is swampy and coastal environments. The Jose Creek flora is more inland. It's about 200 kilometers inland from where the seaway was at the time. It's on, solid ground and more upland. So we might see something different. That was at least a, thought or the hope. Okay, so for the rest of this, we're going to dive into the reconstruction of the Jose Creek member, and I'll show you how we reconstruct a flora and what we found from it, and then we'll kind of at the end fit it back into this environment here. Okay, so the Jose Creek member is really, it's a series of rocks, you see it here, a series of rock layers that preserves an environment kind of on the front of a mountain range, and it's this meandering channel environment. So each of these kind of different sequences of rocks, we have these mudstones, which represent deposition on the floodplain outside of the channels, and you have sandstones representing deposition in the channels. But we also have these ash layers from nearby volcanic activity that preserve across the landscape, scattered throughout this sequence as well. And throughout the whole thing, we have plant fossils. We have fossil leaves, fossil wood, fossil forest horizons, just a really, really nice plant record. Now I'm interested in kind of reconstructing snapshots, looking at vegetation in one slice of time, one landscape. So I focused in on one single plant layer here. And this was an ashfall layer, which is called Dori's tuff. Now, I should mention, I didn't name that layer after myself. The geologist that was writing up the section actually did that, named it Dori's tuff, which for a while I felt really ridiculous about, but now it just seems fun to talk about Dori's tuff. So this is a really nice deposit, other than being named after me. It's an ashfall bed, so this is the result of ash raining down on a landscape and preserving everything that was there. And this ash layer, which is here, lies on top of a mudstone. And this mudstone is from floodplain deposit. So what this tells us is this is ash falling on a floodplain and preserving the vegetation that was here on the floodplain. Now, ash is really cool also because it can be radiometrically dated. And this layer has been dated at 74.6 million years old. So we have a nice date on that. Now, when I first went out there as an eager PhD student to start studying this, we knew that there was this lovely outcrop here on this hillside that preserved fossil plants. And so I go out there with my colleagues, and we decided, okay, well, let's walk this out. Let's see how far this deposit goes. And much to our surprise, the deposit was 1.2 kilometers long. And now Big Cedar Ridge was 4, this is 1, but both of them are unusual in being that big of an extent and in preserving kind of snapshots buried in ash, right? So here's this 1.2 kilometers, you can kind of see it along this panorama hillside. Here's the overview of it right there. And same kind of looking back, here's that ash layer, and it winds around here. Now, this was incredible because essentially what we have is this forest preserved in a moment in time, and you can basically take just a 1 kilometer long walk through this forest. If you sample what's in the rock layer, you learn and you can look through the forest, what was here, what was there, and you basically just explore it through a nice meandering walk. So that's basically how we reconstruct it. So let's dig into that. So how we sample along that to reconstruct it is you dig a bunch of quarries. In this case, I dug 26 quarries along that outcrop. And then from that, counted everything in those different quarries. And we describe these things as leaf morphotypes. And this is roughly equivalent to species, each different type of leaf that you find. And so this is what it looks like as you keep sampling. You end up with a pretty big hole in the ground. And when you're sampling to reconstruct ecology, the important thing is that you count and identify everything. You want to do this in a very unbiased way, so you have kind of true counts of relative abundance. So you pull out all the rocks, you crack them open to expose the fossils, you lay them out, and then you go to work. And so I counted these things by 2 different measures. One was relative abundance, that's looking at the number of leaves of each different type of species, but then also this metric called percent cover. So this is looking more at the relative surface area of each leaf on the rock. And did that for a lot of specimens, a lot of rock surface over those 26 quarries. And I must say, when you're sampling on that kind of scale, this takes a lot of people. No one person does that. And so I had a lot of volunteers, a lot of students came out with me over the years and really helped contribute to this work. And I'm very thankful for all of them. And that's important because a lot of the work is not actually glamorous. So busting open rocks, looking at the fossils, that's the really fun part, but a lot of it is a lot less glamorous. It's identifying and counting things, wrapping them up to take back to the museum. So really important that I had really good teams of people with me along the way. All right, so we did all of that work. Well, what did we find? Well, first let's look at diversity. So we found that this deposit had over 200 different leaf morphotypes, so 200 different leaf species. Over 160 of them were recovered in the census. And of those different types, the overwhelming majority of them were flowering plants. So nearly 90% of that diversity was flowering plants, made up mostly of dicots and then also monocots. So monocots are like palms, modern day grasses and things like that are monocots as well. We also had some ferns and conifers, cycad, a few other things, but most of it was angiosperms. You think, okay, well, I sampled a lot of stuff. Did we actually capture all of that diversity? Well, a way to look at that is to look at what we call species accumulation curves. So this plots the number of species here's morphotypes, as sampling intensity increases. So as we sample more specimens, here along the x-axis, do we find more and more species? Now the answer turns out for ferns, monocots, conifers, and other things is not really, right? We saturated that pretty much as I keep sampling, I keep digging holes, keep looking at things. I'm not finding more ferns, more monocots, more conifers. Dicots, on the other hand, are a totally different story. Every time I dig a hole, I find more, and that just seems to have no end. So we definitely didn't saturate the diversity here, but doesn't look like we would ever if I keep digging more holes. But what it tells us is there's a lot, simply a lot. And I'd say, okay, so those are some great numbers, but what does that mean in terms of diversity in the fossil record? Well, it turns out it's really the most diverse single flora that we know of. So here's the species accumulation curves again, our sample size by, in this case, dicot morphotypes. Here's Dori's tuft, this blue line here, that shows my sampling curve. And here are other fossil floras known from mostly the early Cenozoic. These are other tropical and subtropical floras, so similar climate environment. And you can see that their curves are much lower. Even though some of these sampled even more than I did, they still didn't attain the same diversity. And the really one that's only kind of similar is this flora called the Castle Rock flora. It turns out this is that Colorado rainforest that I was telling you about earlier. So it's kind of in line with this really diverse rainforest that we know of from right after the end of the Cretaceous. Now we can throw, these are the fossil floras, so we can throw some living floras on there to add a little more perspective. So here's added now on here, a site from Brazil and a site from Panama, so really diverse modern rainforest. And this is data basically taken from sampling in those forests from the leaf litters as a way to mimic the way we find fossils. And basically what you can take from this is that the Dori's tuft flora as well as the Castle Rock flora are basically on par with the diversity that we see in some of our really diverse rainforest environments today. So quite striking and surprising. Okay, so what does some of that look like? Well, we'll do a really quick walkthrough to show what basically some of these fossils look like. They're beautiful. So we have some conifers. So here's a nice extinct conifer we call the Gynetzia type, as well as this other really cool one called Brachyphyllum, has these scale-like leaves on these really thick, big shoots. And this type of conifer is found all over during the Mesozoic, but it's from extinct stuff that we don't see today. Here's a gorgeous redwood relative. With cycads, here's a zamioid cycad, and cycads, of course, have this very typical large shrub-like habitat, or habit, sorry. We get into some ferns. The ferns are beautiful. I love finding the ferns. They're all these like lovely little delicate specimens. They're always small. The leaves are just, I mean, they're ferns, they're gorgeous. But they're pretty low diversity and actually kind of rare in the deposit, so we don't find them a lot. And we get into our angiosperms. So first, here's just kind of a sampling of some of the monocots. We have this lovely fan palm called Sabalides. This is a real popular one there called Zingiberopsis. It's a fossil ginger species, basically. And these leaves are actually these really big leaves, but I usually find them broken up. So get these smaller samples like this. All these other kind of little herbaceous monocots as well. Here's one called Pandanites that I really like, these really beautiful strap-like leaves with these mean little spines on the margin. So some really cool things. What's interesting about the monocots is they also seem to kind of cover a range of growth habits. So the Sabalides palm, this would be like a shrub to small tree fan palm. The ginger is this rosette forming large leaved kind of ground covering plant. Pandanites, if you can guess from its name, it's often likened to pandanus grass. We don't really actually know if the habit is similar, but it would be this kind of large shrubby monocot, if you will, as well as these like more kind of herbaceous, smaller monocots as well. So you kind of have this full range of ecologies from the monocots. Okay, and then we get into I'll show just a couple slides, a broad sample of the dicots. The dicots are really diverse. There's so many of them. This is just a page of ones with tooth margins here. Fossil people, we love to divide dicots by tooth or not tooth. Even have some with some spines, but a whole bunch of different types. Most of them, though, are not toothed. They tend to have these smooth margins like this. Here's one that's just been obliterated by insects, so there's a lot of herbivory damage on it, and this is actually really common through the deposit as well. We see lots of hole feeding, little leaf mines, and all kinds of evidence of insects just chomping on the leaves. And even more dicot angiosperms, again, very beautiful leaves. A lot of these also have these really long tips. We call these drip tips. You can see them on several specimens. They often get broken off, but that's an adaptation in really humid or wet environments. So something to keep in mind. Okay, so let's go ahead and move on to, we talked about diversity, but we took all this count data, this abundance data. What did that end up telling us? Well, in a nutshell, when we plot out the numbers of specimens by these major groups, dicots, monocots, conifers, everything else, what we find is that overwhelmingly, without question, flowering plants are the most abundant thing across this landscape. So the dicots plus the monocots just overwhelmingly dominate. Conifers, depending on what metric we use, that number of specimens or the percent cover, come up to 20% of the flora, but overall, we're pretty much in angiosperm land. Now this is looking at them all that whole deposit lumped together. We took all that really great spatial data by sampling in quarries along the deposit. So let's see what it looks like when we look at that. So here's that kind of walk through the forest here. And at each one of our sample sites, I've plotted a bar that shows the relative abundance of the different plant groups. So our flowering plants in the dicot plus monocot, conifers brown, and then our ferns and cycads. What you can see when you look across the deposit is there's actually a lot of variation in the composition, site to site. But still, overall, angiosperms are abundant across the whole thing. And this is an important point because it shows that it's not that we have angiosperms living over here and we have conifers living over here. Really, they're mixed together, angiosperms and conifers, across this whole landscape, across this floodplain. Now, this is, of course, just that broad look at relative abundance by the major groups. I've done this looking at it by species too, like what are all the different species at this site? What about the species at this site and at this site? Are they different? Are they the same? And it turns out that basically we have really high species variability across the deposit. So the species here are not the same as the species here, or different than the species here. And that's what makes up the high diversity of this site. It's what we call really spatially heterogeneous. So as you're walking through the forest, you're constantly encountering new species, different species, different species. This is a pattern that we see in modern tropical forests. This really diverse, you walk to a different place, it's like totally different species. Whereas when you're looking at temperate forests, like our forests like in the northern US, as you walk a mile through a forest, you're gonna see the same things over and over and over and over again. That's a real homogeneous landscape. So it turns out that although angiosperms are dominant across it, it's really still very heterogeneous, and there's a lot of species diversity. Now, this makes sense when we plot out the what we call the rank abundance of species. So for every one of the species we find in the deposit, if you rank them by the most common to least common, here's their abundance. But you see is that there's this distribution where you have a few things common, and then this really, really long tail of things that are rare, not very common. And this is a pattern that we see in these real high diversity, spatially variable landscapes. Now, an important point is that most of these really rare things are dicots. I color-coded in that teal. So we have a few common things and then really, really, really diverse but not common dicots. And interestingly, really like 84% of the morphotypes in the deposit are found in less than 3 quarries, 3 out of those 26. So you're seeing very different things all over the place. But since we do have a few common things, let's hone in on those and see what we're looking at. What is common? Well, overwhelmingly, the top 2 things that we see across the deposit is this ginger, that ground-covered ginger, big rosette-forming leaves, and then the redwood relative, which of course was a tree. Those are the 2 most common, very quickly followed by several others. That brachyphyllum conifer, 2 dicots that would have been trees, the Sabolites palm, and a cycad. So these are the most common things across the landscape. All right, so let's move on to kind of another line of evidence. I showed y'all earlier how there's actually a lot of fossil wood in the deposit as well. And my colleagues have been working on that wood, and there's some insights we can gain from that wood into understanding what the angiosperms and conifers and such were doing in the Dori's tough flora. Well, what they found is that in studying the wood flora, that they have discovered that there's all the way from small to large angiosperm stumps and logs. So angiosperms were forming everything from small trees to really, really big trees. And very notably, this site actually has the largest Cretaceous angiosperm known. So here is a stump of an angiosperm that my colleagues Emilio and Joan are sitting in front of that just goes from here to here. I mean, it's a gigantic tree. So we know that angiosperms were forming some really big trees in this flora. We have evidence of palm wood, and they had everything from large shrub and tree habit. We have big ones and small ones, quite a diversity. Conifers, of course, form some pretty big stumps as well. Here's one sitting right in and sticking up above the ash layer. So what we know from this is that both angiosperms and conifers and palms were forming elements of the canopy. They're kind of all doing it. Now, we also have done from both the wood and the leaf data that I've collected and some paleoclimate inferences. I'm not going to go into the details of it, but basically, because plants have such an intimate relationship with their environment, there's really cool methods you can use to be able to infer things like temperature and precipitation and take a look at seasonality and things like that from features of the leaves and wood. And just a summary of it is that we've basically found that this was a really warm environment. We're in tropical temperatures. There was no freezing. Precipitation was likely year-round, so it's a really moist, wet, and warm environment. And notably, there's a lack of seasonality. So even in wood sections, we don't really see strong regular growth rings in the woods. And in the foliage, there's not really signs of dormant periods in the foliage. So overall, this all indicates that this was a tropical climate, which makes sense given the location of the site and what we know about the time period. So this fits well into what we understand. Okay, so pulling all that together, the growth habits, the climate, everything we reconstructed from the flora, the basic summary is that the Dori's tough flora represents a really diverse, spatially heterogeneous tropical forest with abundant flowering plants that coexisted with conifers in a mixed canopy. And that last part's a really important point, that conifers and flowering plants were really living together in this mixed canopy and not separate parts of the landscape like we've seen in other floras. And of course, this is what we call a really classic example of a non-analog community. And that basically means that it contains a mixture of plant groups that are no longer seen in association today. So like palms and redwoods and cycads living together with a ginger understory. And then, of course, this backdrop of really, really diverse dicots kind of intermixed through it all. So this is an environment completely unlike any of the ones we necessarily have today. Of course, there's dinosaurs also tramping all through there to make it even more fun. But anyway, so this is a very interesting and insightful flora. So let's take what we've learned about this and let's plot this back on this late Campanian landscape in context with these other floras and see what we've learned. Well, So first of all, some really notable aspects of Dori's tough flora in relation to these others is, well, by far it's the most diverse. It's also the first record where we can really show with numbers that flowering plants dominated the floodplain. It wasn't just in channel environments, but they were abundant and dominant across the floodplain. And then the mixed canopy where flowering plants and conifers were coexisting. And so this brings up actually a lot of questions. So why is this flora showing us different things than what we've seen in these other ones? And there's a couple things that come to mind. One, it's, again, further south. Is this part of a latitudinal trend? We know that angiosperms radiated out from the equatorial regions. Maybe this is just a continuation of that trend as they take over, really. Or maybe it's climate. This is also more tropical and warmer than some of these floras that we study up here. Could that be playing into it? And then there's also the habit. This is inland and upland. It's a different environment than most floras we know at the time, which are in these coastal and swampy environments. And really, it could be all of these things. And we don't actually know yet. This is going to take a lot more work as we kind of start teasing out these different factors. But one thing that it really helps us understand is that here at the Campanian, before we get to the very end of the Cretaceous, we do have landscapes out there where flowering plants are dominating. And as we continue forward, hopefully we'll learn more. You know, this was one site, kind of in one slice of time in one place. And there's a lot more to explore to be able to kind of piece together really these patterns of change in the Cretaceous as angiosperms radiated and took over environments, and what that means for the animals at the time and everything thereafter. And with that, I guess I would like to note that work like this, studies, especially extensive ones, only get done with the help of a lot of institutions and a lot of people and a lot of funding. So I want to thank and acknowledge everyone that contributed to these studies, both indirectly and directly, and everyone that came before to set that stage to help understand what's going on in the history of our planet. All right, and with that, I'd be really happy to take any questions that you may have.
Yay! Thank you, Dori. That was fantastic. Oh my goodness, so amazing. Yes, we have quite a few questions. Yeah, so we'll start from the beginning. And this question from Rocio Diana: what are your thoughts about the ongoing discussion on the time of origin of angiosperms?
That is a great question. I'm always careful in talks like these to mention, you know, especially talking about a Cretaceous angiosperms, is that's when we first see them in the fossil record, but it's not necessarily the origin, right? And this has become a hot topic recently. Some new research just came out based on some modeling and dating procedures that kind of push back the origin time of angiosperms to, I forget what the paper says, anyway, far earlier than the Cretaceous. And this comes up periodically because it just is a constant question. When did they originate? I mean, you know, when we see them in the fossil record, it's just when they appear. So we know their origination had to happen before they appear in the fossil record, right? Because it's before the evidence comes up. And the big questions are really just how long before they appear in the early Cretaceous did the angiosperms originate? And I don't really have what would be the saying? I don't really have a strong feeling in the fight about that. Some people can be very, have really strong opinions about that. I think, you know, a general consensus might be that it's of course reasonable that angiosperms were around in the late Jurassic, just kind of really low, but how far that chain goes back before, it's hard to say. And the other thing is, you know, the origin could be well before we would recognize them as angiosperms like we do today. So there's a lot of questions about what would the early angiosperms and the precursors to them that are different than the other gymnosperms, what would those look like? And that becomes harder to recognize. And yeah, I hope that helps answer that. Thank you.
Yeah, that's great. Let's see, I believe the same person had a follow-up question to that. How different would this landscape reconstruction be in South America during the Cretaceous? Are some available?
Probably very different. I mean, if we just look at the world today, right? From place to place, things are very different. And across vegetation types, you have similarities, different species, but similarities in traits and characteristics of vegetation. My guess would be if we had similar age floras reconstructed like this in South America, you might see similar. It just depends on, I think the climate patterns in wherever you are really influences a lot, that combination of climate and habitat. But by no means would this be the only site like this. There's clearly more out there. We just don't have all those data points, I think.
Great, thanks. Another question, this one from Tiana Raymond. Are all the samples from your quarries deposited in a natural history collection, or do you select a representative specimen of each morphotype?
That is a great question. Unfortunately, I couldn't bring the whole hillside back to the museum. I ended up collecting representatives of each specimens. So a lot of samples are still out there. We did a lot of counting and tabulating in the field. What I ended up doing was bringing back the things that helped with describing the species, or that were really well preserved to where we could get really good measurements of different traits from them later for studies down the road. So the things that I didn't collect were specimens that were pretty partial, or just we had so many of them already that it didn't really add to the knowledge base very well.
Great, thanks. That's our collections manager here at Brit.
Okay.
That question.
It was a lot of rocks.
I can only imagine shipping costs.
Yeah,
right?
we didn't ship. I drove them back in an SUV. So each time I came back, like, oh my gosh, we were low riding on that drive back from New Mexico to California, because they're also, this layer of the rocks happen to be really heavy. It's a very dense ash. There's a lot of silica in there and things. And so it's just a really, really hard rock. So it was some very weighty loads.
All right, we've got another question. This one from Nathan. Did you break down your dicots in the herbs, trees or shrubs, climbers, or uncertain? And then he had another one, and also distribution of leaf sizes.
Yes, so some of that work is still ongoing. As Nathan also works on fossil plants, and as he knows, it's very difficult to actually break down into different habitat groups within the dicots. But that is some things that were ongoing. So, I have hypotheses about each one, but I don't have that data like ready to go yet. And same for leaf sizes. So surprisingly, despite kind of all the inferences of this being a fairly wet and tropical environment, if you will, the leaf sizes really aren't that big. They're not giant leaves. I forget the size range is kind of offhand. I mean, we do have a decent, range of sizes, but overall they're not very big.
Okay. Let's see. From Deanna again, following the question of Nathan, what proportion of different dicot organs did you find? Mostly leaves? Mostly fruits?
Mostly leaves. Like I would say, I don't actually have numbers on those proportions because I didn't necessarily count all the reproductive stuff in the same way. I became interested in the reproductive stuff later. We do have a lot of seeds and fruits of pretty varying sizes, and had a student who's now a PhD student that has been working on describing that diversity of reproductive morphotypes from the dicots. So that'll be some work coming out soon, but it's actually really diverse in quite a range of like sizes and types, and probably dispersal habits and stuff. But most of what the deposit was leaves, a lot of leaves and kind of stems, but definitely still a decent amount of reproductive material. Not as many flowers as I would like, but there were some flowers still too.
This is kind of a similar question. Are fungi ever found? This is from Ashley Bordelon. Are fungi ever found, or are they too fleshy to preserve in this deposit?
In this deposit, I don't see fungi, but I know in other deposits they can preserve really well. I think some of, in the wood samples, you can see evidence of some of that in the permineralized things, but I don't really see it preserved well on the leaves.
Okay. Let's see. A question from Mario Corriro. It's surprising to me that the cycads were so abundant, even though still a minor component. Do you have any ideas of the precise affinities?
No, and it looks like there's only 1 in to maybe 2. Me and Gary Upchurch argue about the second one. But it seems like a zamioid cycad. I haven't dug into it much further than that, other than kind of typing it out. I should get in touch with you about that since you know a lot about cycads. We have a lot of paleobotanists in the house, it turns out.
Is it a small world?
We're a pretty tight crew.
Let's see, we have a question from Alejandra. How many new species of fossils have you described from there? Are there affinities with other closer fossil sites?
Yeah, so sites like this, and this is kind of the next kind of long-term phase of this. I will probably be describing species from this for the rest of my life. It's a lot of work to go and actually describe and really justify the affinity of each fossil. And with the dicots, it can be really hard because the leaves can be really difficult to kind of ascribe affinities to. So I'm kind of tackling this really from the low-hanging fruit on. So I've started working on the conifers and the monocots. And we'll start, progressing from there, but it will be a very large proportion of them will actually be new species. The big question is whether or not we can actually put names on them all. And some of this just comes down to being able to effectively compare these fossils from this site with other fossils from other Cretaceous sites that have been describing leaves as morphotypes. And yeah, it's a big task to get all of that done, but it'll be kind of ongoing, and there will be a lot.
All right. A question from Janet Riley. Is the ash that you refer to due to volcanoes in the area? Yeah.
I don't know how close the volcanoes would have been, but yes, they would have been far, close enough that you have ash raining down. But this ash is a really fine grain ash, so we know that it's far enough off that you're not right beside it. It's this very kind of, just nice fine grain, not the big chunks that you get if you're closer to the volcanoes. But yeah, and I think from what I understand of it, they would have been to the west.
Okay. All right, we have a question from Max Portman. Are there other clues you would expect to find from surrounding geologic formations that would help reconstruct the climate during estimated angiosperm origin?
Yeah, so pretty much, because of that relationship between plants and climate, anyone that's working on large floras is generally also using the data to do climate reconstructions for that spot. And so we have a lot of them. And then that kind of goes along with data that we know from other paleontologists that, for say, work on marine stuff and can do cool things to get sea surface temperature estimates and things. So we actually have, a pretty decent idea of the climates through the Cretaceous and in different areas, and modeling helps that as well. So we know that kind of during this time, during the early Cretaceous, we did have warm periods. And within that, overall it was warm. And within that, you have periods that kind of go up and down, peak warm times, and kind of fluctuates. But yeah, it's a pretty good idea of it, from when you talk about the global data of climate reconstructions.
Thanks. A question from Manuela Del Forno. Do you find many animals too? And if yes, which ones and what do you do with them? Do you have collaborators for those?
I try and avoid the animals. That's just going to get me into trouble. No, so I don't actually find them within the layer that I'm studying. So I stay pretty focused in on working with this plant layer. We do see, and I mentioned we see the insect damage. So that's kind of an indirect evidence of the animals, because it's what we call trace fossils. It's not the bodies of the insects themselves, but it's evidence of their existence. Mm-hmm. And what's cool is those insect damage actually, that field of study is kind of a hot field of study, and they can actually use those insect damages to even kind of learn about what types of insects were there. So there's a cool indirect evidence of at least the insects, the ones that feed on plants. But also there is work that's been done in the formation and the general area that talk about the animals. So I know there's like some hadrosaur bones, there's Triceratops. I don't know if it's actually Triceratops or some other ceratopsian species. But, you know, the typical dinosaurs that you would expect at this time, they see some of them in the formation and from surrounding formations. So you get kind of a view at what large animals were tramping around then.
Very cool.
Yeah, other people work on that, not me.
Were they out? I mean, during your times, though, do you have a kind of multi-species group? Like you're focused on your plants, but is there someone in the next hole over working on something else?
Not that close. You know, there is so much work to be done and not nearly enough funding and people to do all of the work to learn about this history. So, you know, I work with a team of paleobotanists, really, you know, my colleagues that have been working on the wood flora, and had worked a little bit on some of the other leaf floras in the area before me. And that's, you know, a decent group already, but, you know, there hasn't been too much activity on the dinos from this exact site. Now in surrounding basins, like by all means, there are some crews that have been doing some fabulous work on the dinosaurs and floras, but this one's kind of a little less known, I would say.
Let's see, another question from Nathan. Are there nearby shales with other fossil leaves, up section or down section?
There are, not as well preserved often. So there's leaves preserved in the sandstones as well. Of course, those are, you know, less nicely preserved at some pretty coarse sandstones. The ash layers tend to have leaves in them, and there are other ash layers. The hillside itself is not that big, so it gets hard to kind of correlate sections across big distances. Like this ash layer probably exists other places on the ranch that this was at. It's just hard to correlate with certainty, but there are, there's a lot of plant fossils out there.
For us newbies, does up section mean like younger?
Yeah, up section will be younger and down section will be older, typically, unless all that got flipped over, but typically it will be, up section will be younger, down section will be older.
Great, thanks. Let's see, we have a question from Alvaro Javier Perez Castaneda. Are there any magnolia fossils in your study site?
There are definitely fossils that belong to the Magnoliales, so the bigger group. Now, I wouldn't say Magnolia as a genus per se, but I tend to be hesitant about ascribing affinities to the Dicots in general without a lot more work.
Great. Oh, all the questions have been interesting, but this one I've not thought about this. This is from Natch Azure. Given what you know about these plants and their relatives today, what do you think some of these flowers smelled like?
That's really interesting. Right? I can honestly say that I don't think I've ever, of all the many questions I've had, I don't think I have ever wondered that as I've been out there. You know, I would love to think that they smelled glorious. But I really have no even educated guess on that. I will say that the flowers from this deposit, the ones that I found, now clearly, like we've found a couple of different flowers. Flowers are really rare to find in the deposit, partially because, you know, things have to be in bloom when this ashfall fell. You know, a whole lot of other things go into whether or not you actually find them. But all the ones we do find are really tiny. They're like really, really tiny flowers. Yeah. Don't know why, but I can think of some really tiny flowers today that just smell amazing. So I really have no idea. What's fun to think about, though.
Yeah, I would imagine the smell would probably be related to what the smell was for. So if it was attracting pollinators, which pollinators were around at that time. So gotta go hang out with the other colleagues to
I need to talk to some more insect people, see what their thoughts on that would be. They'd probably have some better thoughts on it.
Yeah, let's see. Did we miss any questions in there? There's some more coming in.
These are fun questions.
Lots of thanks and wonderful lecture. Let's see. Someone says, Alexis Rastier says, for the study of the evolution of angiosperms, it could be very interesting to look at the paleodiversity of flowering plants at the beginning of the Cenomanian and Turonian stages.
Oh, yes, those would be amazing. Luckily, some of my work is going to take me into studying things here in Texas, which gets a little older. We have some really nice Cretaceous sites here in Texas, so I'm eager to kind of look back further in time. But yeah, there's just, we have a really big record from the Campanian. That's why so much focus on that time slice. And we've learned a lot about it, but it's definitely will be nice to get a deeper understanding of the slices before that.
Yeah. Someone asks, have you found anything similar to Solanales? Someone's really interested in seeds and fruits.
Nothing that's definitive. Most of, unfortunately, because this is mostly an impression compression deposit, so that everything's kind of flattened and smushed, it makes it really difficult with the fruits and seeds to kind of tell really definite affinities if there's not some structures projecting or really obvious things, because it's like a bunch of smushed balls, if you will. So it's a really tricky thing to work out in deposits where they're like three-dimensionally preserved. You can like slice them open, see all the anatomy, and do cool things like that. But we're kind of still in that earlier phase of trying to ascribe real affinities or identifications to the fruits and seeds. And not sure how far we'll be able to take them all.
Yeah. Let's see, we're getting close to our time here. Actually, we're a little over time, but there's just a few questions remaining. I think you mentioned that this site is on a ranch. Is it a protected site?
It is a privately owned site.
Okay.
It's been set aside for conservation, but it is privately owned. So, I don't know what the future will hold, but we'll see.
This is an interjection on this kind of same theme. So, does the landowner own the fossils?
They own them until they're donated to the museum to reposit. So you work with in cases where you're working with private landowners, you work out an arrangement to where you can permanently reposit the fossils, because it's important that the specimens be preserved for future generations and be available for science and education and things like that. So that's a matter of making agreements with private landowners.
Right.
That's one of the things that comes up as you're working in areas like here in Texas, most of the land is private land. And so really you're at the mercy of whether or not landowners will allow you to work on their land. They own the fossils that are on their land. So, it's up to their generosity and kind of how they feel about, really kind of preserving science for the future, to whether or not you actually even get access or the ability to work on them. Because you can look at them, but if you can't really put them in a museum and preserve them, then it's hard to actually do any science with them.
Right. Might be out of your wheelhouse, but would that be the same? Would that still be true if they were like cultural artifacts?
So I know there's some different laws around cultural artifacts than fossils. And I don't know all the details of them, so I don't want to misspeak on it, but I know there's a lot more strict laws about that. But I mean, it will vary by state too, what the laws of any state is in governing those things.
Okay. Let's see. I think we have two more questions. Do you have time for two more?
Yeah.
Okay. All right. So from Alexis, did you know if some abiotic parameters, such as decreasing atmospheric CO2 content during the Upper Cretaceous, could have allowed the expansion of angiosperms probably gymnosperms and filicophytes?
Yeah, I'm not sure. There's a lot of different ideas about kind of the way that the competition between these different groups kind of went throughout the Cretaceous and what the advantages different ones had on the other. Quite could be a combination of things. Angiosperms kind of, they're able to attain photosynthetic rates and do things that the other plant groups haven't kind of gained that ability. They just weren't able to photosynthesize as efficiently, essentially. But yeah, I'm not sure if we could say, that declining CO2 or anything like that necessarily allowed that or caused it, but there could be quite a few things that interplay to help out. Thanks.
Okay, and one final question. This is kind of a funny one. considering I'm in Austin dealing with cedar allergies, sweet cedar in quotes, how bad do you think allergies were back then? Or if dinosaurs had them?
It's kind of funny to think about dinosaurs sneezing, right? Like, that would be so messy. I don't know. So what I would just venture to guess for fun, right? We still had a lot of conifers around then. And conifers, I mean, are wind pollinated for the most part. So you probably had a lot of pollen all over the place. And a lot from the family Cupressaceae, which is what plagues you in Austin. So.
Yes, definitely. All right. Well, if there's one thing that Texas kind of paleobotany is known for, or is really like a prime example of on a global scale, what would it be?
Oh my goodness, I'm like drawing a blank on that. So, I mean, it has some early deposits. A lot of Texas paleobotany, it really hasn't been extensively studied in recent years. So, I mean, it's really time to get Texas paleobotany into the limelight. But there are some formations like the Glen Rose Formation and things like that are known. I can't say that anything's like sticking out in my head as it being like famous for this thing. Dinosaurs are much better known from Texas than the fossil plants are.
Yeah.
Currently,
right. Well, it sounds like it's good job security. You've got your niche, right?
I would like to think, yeah.
Yeah. Well, Dr. Contreras, is there anything you'd like to pitch coming up for the Perot?
I didn't prepare any pitches. I'm just excited, as we move our program forward, we're going to be focusing a lot on Texas, and that's really exciting. Not only because it's our backyard and whatnot, but I think it's going to be really great and interesting for our community. And I'm hoping we can continue getting people around here jazzed about fossils and about Earth history and kind of take that forward. Yeah.
Is the museum open right now?
Yes, the museum is open, limited hours, so we're not open every day. And we do encourage buying tickets in advance, but we're open to limited capacity, which means it's kind of a really nice, not crowded way to tour the museum and see everything kind of on your own time without it being a madhouse. So, yeah.
That's great. Well, thank you so much for being with us here today and sharing your research. It's really exciting. We hope maybe after you describe a couple new species, we'll have you back and you can talk all about those, right?
Great. Well, thank y'all very much. I appreciate everyone tuning in. This was really fun.
Thanks. For those of you still on the line here, I want to promote our next talk. It's actually part of a new student research series, and this will be at a Rare Friday talk at noon on February 19th. It will be topics on Texas aquatic ecosystems from two graduate students, Katie Vasquez and Megan Harlow, both from University of North Texas. And then after that, I don't have a little slide for it, but after that, our March Lunchtime Lecture on March 2nd will be a presentation about the Shirley Tucker Herbarium at LSU. So presented by Laura Lagomarsino and Jennifer Cluse. So we hope you'll tune in here on the YouTubes to join us again in March. Oh, and also that February 19th talk. All right. Thanks, everybody. Take care.